So I've just been told the microphone has not been on this whole time. Everyone just is really good at projecting. So I will also be good at projecting. My name is Kathy Clapridge, and I'm a professor of biomedical engineering here at Boston University, and I am also the scientific director of the DAMP Lab, which means for the last year that I write a lot of grants, and I also was in charge of soliciting and helping choose the pilot projects that you'll hear about, 3 of them today and 3 of them tomorrow. How this section of the workshop is going to go is I will introduce, in some cases, the PI of the project, who will then either give the talk or introduce the graduate student or postdoc who worked most closely with the DAMP Lab, because we want to try to get information, closest to the ground information about how working with the DAMP Lab actually worked hand in hand with the people that worked with our staff and in the laboratories. And then tomorrow we'll do the same thing with those 3 talks. Okay. So the first talk is being presented by James Robson, who is a graduate student in the Alex Green Lab at Boston University. Alex Green is a professor of biomedical engineering, and he is not here today, so I get to introduce James, which is great because James used to be in my lab when he was an undergraduate. And you also have seen James in some videos because he was a newly minted graduate of Boston University in the spring of 2020 and was one of the summer students who helped us put together the assay that went onto the robots in the summer of 2020. So he's been with part of the DAMP Lab since the CTL, and which became part of the DAMP Lab since the very beginning, in those scary times when we were like staying 6 feet apart and wearing masks and having meetings outside and so on. But since he became a graduate student, he has moved on to other more exciting projects, and he'll tell you about one of those today. So, James.
Hi, everyone. My name is James. I'm a 3rd-year biomedical engineering PhD candidate in the Green Lab. And as Kathy just introduced, I started, my undergraduate research, and then right after I graduated, automating kind of these diagnostic workflows. And as I started my PhD, I was really interested in looking at ways that we can screen nucleic acid devices for diagnostics, for broad synthetic biology applications, and the DAMP Lab and the service that in this pilot project that I'll talk to you about was an excellent way to take this automated design that the Green Lab kind of is an expert in, and then implement it in really high throughput ways so that we can analyze how a lot of these nucleic acid sensors work and how they perform. So nucleic acid devices have been built for a variety of different applications, and I kind of want to talk to what I mean by nucleic acid devices. So in a lot of applications, they are point of care tests for different disease states. So as you can see here, the snipers that was released in Cell in 2018 is a colorimetric point of care test for Zika and other viruses, where you can have single nucleotide point mutation specificity for different diagnostic applications. Additionally, these types of nucleic acid sensors can be combined into complex logic circuits for in vivo synthetic biology applications. So there are a lot of different device architectures that we can come up with, that we can design, and that we can experimentally test in silico. So using different computational techniques, we can come up with a lot of different device architectures, different nucleic acid interactions. We can model these using thermodynamic parameters, and we can come up with incredibly large data sets. I think at any time I have a couple 100,000 designs that I think that I want to test or that I come up with that I just can't test experimentally. So in this design, build, test workflow, I can come up with a lot of different designs, and then building them here is a real bottleneck. So being able to synthesize these designs so that we can test them and assess performance is a really limiting factor in the design, build, test cycle. And then additionally, what I want to say is that when we do test these designs, as you can see here on the graph, a lot of these have very little or minimal functional output when we do test them experimentally. So when we're looking for, let's say, fluorescence, right? We want to give you a colorimetric readout and say, if you have COVID, for example, this sample will glow green. Down here on the bottom right, you can see that a lot in the red, a lot of these tests have really high functional performance that you can have this diagnostic output, but a lot of them aren't successful. And so in this type of workflow, the design build test cycle, we iterate through it a lot. And so having an automated workflow so that we can expedite this process would significantly impact the ability to screen for these diagnostic tests for these synthetic biology applications and be a really powerful tool for our lab and many labs in particular. And so today I'm going to specifically emphasize the build process here, where we go from single-stranded DNA to fully cloned plasmids and isolate that DNA. So this screening is very laborious and time-consuming. So when we approach the DAMP lab, we have all these designs that we want to test, and oftentimes these designs are just ordered from IDT as single-stranded DNAs. And getting from that single-stranded DNA to a fully cloned plasmid takes a lot of work. And so first you have to run PCR to make them double-stranded, then you have to go through the molecular cloning process, and then transform bacteria to amplify those successfully cloned plasmids. And then you have to isolate those plasmids that you've successfully made, and then finally verify them with sequencing. And so this whole process altogether, when you're running many, many samples, takes a lot of time. It's very easy to do in very small numbers, but it can get exponentially more time consuming as you increase the number of samples. And so to dive a little bit more into the process, the first step here is PCR. So we order these ultimers, these single-stranded nucleotide chains from IDT, and then we create double-stranded DNA. Then you take that double-stranded DNA and you insert them into a destination vector, so a plasmid that you can then amplify through bacterial transformation. So you take this plasmid that you synthesized, you put them into bacteria, and then you plate them so that you can select for a successfully cloned colony that is indicated by a specific resistance marker. And so the hands-on time for a lot of these processes increases the more samples that you do. So for instance, preparing the PCR, you have to manually pipette all these samples from one plate to another with master mixers and primers. Same with molecular cloning. And then bacterial transformation also is very time consuming, especially with the plating part. So you're able to perform all these processes on each individual sample, but then taking all of these newly constructed constructs and then plating them out on plates so that you can select for colonies is a very time-consuming process. On the second day, after you select for these colonies, you have to pick those colonies and grow them up in culture so that you can amplify that plasmid. Additionally, you have to prepare that plasmid by then taking that DNA that you've put into bacteria and take it out. And so this is called miniprep. And so when you're doing this process, there are many incubation steps. There are many hands-on labor steps that take a lot of time. And then finally, you have to send this out for sequencing to verify what you've done, the constructs that you've made is actually correct. And so the Green Lab turned to the DAMP lab and said, hey, we have the ability to create all these different designs and we can order them, but we have this bottleneck in building them and testing them. And so we sought to automate PCR, the cloning, bacterial transformation on day one, and then also the mini prep, isolating that DNA from bacteria on day two. And there were kind of two key metrics that we wanted the DAMP lab to improve upon. The first was that manual cloning, as we performed this whole process, around 2/3 to 3/4 of all the colonies that we sequence actually sequence correctly. So while we're able to do this manually, the fidelity of this process that we perform isn't always, perfect. And so we want to improve this process with automation to see if we can improve the successful clones that we generate throughout this process. And then secondly, we most ideally want to reduce the hands-on labor in these processes so that we can get back to designing new devices, new diagnostic tests, and so that we don't have to perform a lot of these assays manually. Okay, and so ultimately, this automated nucleic acid device screening will allow for us to screen a greater number of devices in higher throughput. As I said, as I started the presentation, a lot of these devices we test have little to no function. And so if we can tease out those that are functional, and then we can iterate and improve this design build test cycle, then we can improve their design and create better diagnostics and more complex synthetic biology systems. So I think the DAMP lab has been really instrumental in automating these processes and significantly reducing these times with the Hamilton Microlab Star robot. Specifically, you can see here that they've successfully set up these PCR reactions. You can see the gel electrophoresis image up here on the right, where we confirmed the PCR. And then down here, we have a representative plate, a 96-well plate, where we've performed this whole automated cloning workflow, and we've plated 96 samples on a single selection plate with pretty good fidelity of these clones. So moving forward, we wanted to see how this benchmarks with the manual workflow from preparing manually PCR, Gibson, and transformation. And comparing the manual to automation, we saw a 76% decrease in labor time for the user from the manual process of PCR, Gibson, and transformation to the automated process. And this was kind of the key goal here, to reduce the time that it takes for the scientist and the researcher to perform these different workflows. And so it was really great to see that this significantly decreases our time to perform these laborious tasks. So the day 2 processes are still in progress. I'll talk about this in a second, but one thing that we did not kind of identify as a bottleneck was the colony picking and culture inoculation process that we did not seek the DAMP lab to automate. However, we did seek to automate the plasmid preparation process, and the team is still working pretty diligently in order to get that assay functional. But in order to prove that this cloning that I just demonstrated was correct and that this day 1 workflow worked successfully, we decided to take these samples that the Hamiltons pipetted on the agar plates and then complete this process manually in order to verify that these plasmids were successfully synthesized. And so following manual plasmid preparation, greater than 90% of the samples that we selected for were correctly sequenced. So after performing this manual mini-prep process, 32 samples from the 376 total that were cloned were selected for verification, and then 29 of those 32 came back. Additionally, I would like to note that down here on the bottom left, the pairwise identity, meaning the sequence reads that match to the original plasmid sequence, is 100%. So these sequencing reads that we got were really good quality, and there were no mismatches in the 29 samples that we did send out for sequencing. So the PCR cloning and transformation have successfully been automated, and really key, greater than 90% of the samples were successfully sequenced. And one of the key goals here was reduction in manual labor time, and there was greater than 75% reduction in manual labor, which was a really key metric for our lab, so that we can move through screening these devices much faster and much more efficiently. However, moving forward, there are several other bottlenecks that must be addressed. I mentioned the colony picking and culture inoculation earlier because we realized that although we're able to run through the PCR cloning and transformation process for a lot of these samples, while we're able to do that and we have all of these colonies, it took greater than 45 minutes for 4 technologists, including myself, to pick these colonies and start culture inoculations. So that moving forward would be a significant bottleneck to kind of address so that we can expedite this process further. As I said, the plasmid preparation is still in progress. So getting these purified DNA samples from all of these clone constructs is really an essential part to performing these experiments and moving forward. And then finally, one thing that I'd like to mention is that we outsource all of our sequencing here to nearby vendors in the area. So possibly bringing this sequencing capability in-house to BU and having a sequencing core on the Charles River campus would be significantly beneficial. And also reduce time from sample, like I said, single-stranded DNA oligo to results. So fully cloned, sequenced, verified plasmid. And I think having this whole process in-house would greatly expedite and make screening of nucleic acid sensors or devices across labs on the Charles River campus much easier. I'd like to acknowledge Courtney, Dave, and Molly specifically, who were key in getting this pilot project working. I'd like to acknowledge my NSF DRFP fellowship and then the SB2 program here that fund myself. And I will accept any questions. Yeah. Yeah. I can also answer kind of what our lab does. So our lab right now currently has MinION, Oxford Nanopore sequencing device. I run a lot of high throughput screens as well that I didn't touch on here, where we kind of pool a lot of these constructs together, and I run NGS sequencing on them. And I think that's a very affordable in-house option that could be leveraged. But I'm sure Kathy is more.
Yeah. Very seriously at purchasing the newest one. That should have that within 3 years.
Yep.
So that would then complete this and a lot of other projects. So it's still some uncertainty, but looking good.
Yeah.
Yeah. I'm curious about the missing part discriminate. But you seem to show a 96-well format with on.
Right.
Is automation up to
that?
Yeah.
And in the past, I have definitely created the
Oh, yeah.
Well, the footprint of 96 well, right? 9 millimeters around. But the image showed multiple.
Yeah.
So you take on a permanent class within a 9 millimeter.
So there's some coding involved with it. Basically, they did kind of like the next quote of it, kind of like putting the changes are based on the revision.
There are other commercially available automated platforms that perform this task too, like QPIC.
I think what he's asking is, are you choosing size from within 196 well long?
Yeah.
But I mean, it's a fun problem.
Yeah, for sure. Yeah, definitely.
For the sample that failed, so I guess like what kind of information were you thinking about when you like go to the phonics or like sample tracking to help kind of mediate?
Yeah, so the samples that I stated were not sequenced correctly were actually just background colonies for non-correctly linearized plasmid. So in this process, in order to prepare plasmid to accept another DNA strand, you have to digest that or perform a PCR on that plasmid in order to linearize it. And so a lot of the times when we put this plasmid into this kind of workflow and it doesn't work successfully, it's either because that plasmid wasn't digested successfully and then carries through as background in that next step, or there are a number of other processes that might have happened. But in this case, we'd have to just make sure that destination vector that's put into this process is fully digested and fully linearized prior to the process starting.
One last question.
Just curious on the antibiotics question.
Great question. So within this workflow, there were actually two sets of plates, two different plasmids. One was medium low copy number, one was a high. One had kanamycin resistance, the other had CARB resistance. So there were a number of different copy number plasmids that we assessed here, and then different antibiotic resistance as well. Yeah.
Let's thank James.